So myself Kumar Pathak, and here I am going to talk about my research, whatever I did in several days. So my paper that is published in Plant Physiology, having stating that regulation of squalene biosynthesis in plant development by elongated hypocotyle in response to light in a craft of Cystidiana. So basically, let us start with the the introduction part. What is secondary metabolite? So basically, all these secondary metabolites are not essential for the primary metabolic life processes, but they have been very important for mitigating biotic and abiotic stresses, because as we all aware that when plant goes under the specific condition under the stress, what will happen? Two things is here: one the biotic and one is abiotic. So the biotic and abiotic leads, whatever the stress will come, it will produce ROS, and then ROS will leads to the production of antioxidant or the defense gene that can help the plant to stay strong during the stress condition. So I will subsequently, because today's lecture is on the biotic and abiotic both, so I will discuss in subsequent slides. So as we all aware that these secondary metabolites have been classified into the several clades of secondary metabolites like alkaloids, phenolics, terpenoids and phytosterols. So basically, this lab where I am working currently works on flavonoids and terpenoid biosynthesis and other intermediate products. Even we are working on alkaloids in nicotine and many more metabolites. So in my case, I will discuss about the terpenoid. Terpenoid basically synthesized in cytosol as well as in the mitochondrial chloroplast. Even in the perioxysome also, this terpenoid clade takes place, basically MEP pathway. So MEP pathway and MBA pathway happen in cytosol, which basically govern to the acetyl coenzyme, and then subsequently it forms farnesol and then the sesquiterpenes, triterpenes, and then the side chain even in the it clades into the phytosterol biosynthesis. So but MEP pathway basically deal with the chloroplast biogenesis, how this the DXS enzyme is a key enzyme of MEP pathway, which, and in the case of MBA pathway here is HMGR. So I'm not going to give you the detail about the MBA and MEP pathway. Just I'm highlighting their importance, because even you will see the side chain of chlorophyll and plastoquinone being synthesized by MEP pathway. So squalene have precursor molecules, and a broad spectrum molecule can be used from so many things. You can use this secondary metabolite as the biochemical marker for identification of the cancer even, and it have the human health benefits also. So let's talk about how Hi-Fi being involved in this whole MEP pathway. So I have started earlier that DXS and DXR is an important enzyme of MEP pathway, which basically govern for monoterpenes and diterpenes and hemiterpenes. But if you will come to know about the acetyl CoA, which is a primary product of after glycolysis, the Krebs cycle start the Kelvin cycle. So acetyl coenzyme is very important the product for the primary metabolism. So this primary metabolism is also involved in the secondary metabolism. So this is the mevalonate, and in the subsequent clade it's from farnesol to the SQS, which is the key gene where we are working currently. So squalene is very important, and then SQE. So what is my assumption that in the due course of evolution, this part is being under the negative control of with the aspect of the light, and then downstream pathway, because cholesterol and many more factors again is very important for the plant, and light regulate this gene in a positive way. So that's very interesting to know that how this pathway is being involved. So let's talk about my gene and its promoter. So when we scan this 1.9 kb of the promoter, we got the several G boxes, which is well known to interact with the light. So basically I will not explain the same pathway. So just think that what is happening on the SQS expression between light and dark and subsequent in the transition zone. Whenever the high 5 is functional, the SQS expression and content is decreasing subsequently, and then in a time dependent manner you can see here that the expression of SQS 1 is going down. So what does it mean? It means that SQS expression is going gradually decreasing. It means high 5, whenever the functional high 5 is there, the expression goes down. So to further validate, this is a whole thing has been carried out in the white light. So what will happen in the different light qualities like blue light, far red light, and far red light, and the red light. So you can see here that the white light, blue light, red light, and far red light is showing some kind of sync that SQS expression is always going down. At the respect, you will see the high 5 expression is going up. So it means that the light and SQS have a negative correlation under different light qualities also. But if you will see the content level, why I was explaining previously in the pathway diagram that how this pathway is being controlled under the positive control and negative control, because plant have evolved themselves in a way that they act in a way that this pathway, this is choline and upstream pathway, have a negative correlation with the light, and if you will see the cholesterol part, it's increasing in different qualities, right? So to further validation of this hypothesis, what we did basically, we have chosen the high 5 modulated lines, one high 5 mutant and one high 5 OX. You can see here that the SQS content and expression is going higher in high 5 mutant, which I have earlier suggested that in mutant line SQS content is in the dark condition it is increasing as compared to the light. So high 5 mutant is as like a dark condition, and high 5 OX in the means high 5 is more. So you can see here that same type of significant result we got, that whenever the high 5 is functional, SQS expression is going down. In high 5 mutant line, this is non-significant result, is indicating that whenever high 5 is not available, SQS activity is not being that much modulated. So this is all the hypothetical answer and expression data. So this is not the proper evidence. We have to show some molecular evidence. Yes, of course, G box is available on the cis regulatory element, and then high 5 is coming and interacting with that and regulating the pathway. Okay, so for that, first of all, what we did basically. So okay, so I will keep here. So this is the 4 G boxes which I have earlier stated in the cartoon, and you can see here that all of the 4 G boxes is interacting well. So you can see here also that through the Y1H assay we have concluded that the whole fragment is interacting and giving the strong interaction. But this is not evidently the perfect answer to tell that yes, this is interacting or not. For that we have generated no ACGT box construct where the G box is mutated and then cloned. Even we have what we have did that we have taken the fragment without that ACGT functional core element, and then we performed Y1H, and we got that whenever the G box is not available in the system, the interaction got abolished. So it means this is very specific to the G box. So for further validation, we have purified that protein and then performed IMSA and followed by CHIP. So fragment 2 and 3 have a significant, the interaction enrichment, relative enrichment is maximum in fragment 2 and fragment 3. So this means that it's all data correctly evident that yes, of course, high 5 come into the picture. This is the cis regulatory element. High 5 come and binds and then regulate. So this is the in vitro study. So for in vivo study, what we did, we have cloned this fragment in high 5 mutant and high 5 overexpression line. So you can see here that in the light condition, if you will see the light, so this is the dark gas and this is the light gas. So of course in light condition the gas accumulation was relatively reduced as compared to the dark. But in high 5 mutant, this is not as such. Whatever is in dark, this is same in the light. But whenever the high 5 is functional, interaction got drastically changed. So this is the GFP because whatever we generated our knockout promoted lines, they are in the fusion with the GFP also. So this is again that whatever I have stated earlier, that this is very important to understand the phenotype. So it's important to understand what will happen when we generate knockout mutant of H2S1, because this is a key gene, and downstream cholesterol biosynthesis is important. When upstream element is got mutated, what will happen? Whole phytosterol pathway is being hampered. As a result, plant growth development drastically affected in the mutant background. So one interesting story we have, still I'm wondering that what happened to these plants basically. So when we compare SQS CRISPR in high 5 215 and high 5 215 alone, so you can see a very high hypocotyl length is here. But in the mutant of high 5 in that background, SQS mutant in high 5 mutant background, you can see as compared to the wild type, even this the neck, hypocotyl neck is shortened. So this is a fantastic story. We don't know why this is happening in the plant system. We are planning to do transcriptome analysis, and then we will work on this prospect also. So this story is very nice, but the thing is, what will happen when we supplement exogenous squalene to this mutant, whatever we have generated to the CRISPR strategy. So when we generated this knockout mutant, and then you can see here that this is the control, and when we supplement exogenous squalene to the plants, subsequently it's the root length phenotype is increasing, and even the plant growth promotion is also increasing at up to the 300 to 500. But 500 nanogram per microliter and further subsequently, it's again start reducing the phenotype. It means what? It means the higher concentration of squalene is also lethal for the plant. So again we want to check in the dark, so that not much significant change be observed in the dark grown seedlings. So what is the importance on abiotic stress? A reviewer asked me. Initially we thought that this is more than enough data to show that yes, of course, high 5 coming and binding and regulating the expression. So reviewer asked me to show some the physiological significance of your study. For that we have planned the different studies. So initially we have grown the seedling on the mannitol. This is the mannitol. So on mannitol we have grown this the various overexpression CRISPR and high 5OX in this background, and we come to know that this background SQS-OX in high 5 2.5 has a drastic change over and under the reduction. You can see here the reduction percentage in the this background. So what is happening here, that already high 5 mutant here, so squalene is higher, and again we are increasing the squalene content through overexpressing this gene. So it is known earlier in Rice's paper, in very nice story. I forgot the name of the author to mention here. That they have nicely shown that how this RNA line of rice is very drastically negatively regulating this whole scenario. So to further validate that hypothesis, we have give hydration dehydration type of response we have generated. So you can see here that when we starve plants from giving waters, so CRISPR line is behaving in a better way as compared to the overexpression plant. So overexpression plants drying earlier, and it's showing when we try to recover this process. So high 5 overexpression plant is not able to recover in a prominent way as compared to the CRISPR plant. So this whole story stated that yes, of course, this gene have the great control on the abiotic stress. So we again check this impact on the temperature basis. So both the gene works in a synergistic way in comparison to the temperature stress. So for further, reviewer have asked to mimic the soil environment to make them some low phosphorus, low sulfate, minus iron. So this story has been showing that SQS under the different the low phosphate, low sulfur, and minus iron condition, showing the negative correlation as compared to the high 5. In high 5 expression is going increased in all the above the low phosphate, low sulfur, and minus iron condition. So if my iron is minus, then what is happening with the phosphate? I don't know. I want to ask from you. Basically, this is not my domain. Basically, I try to correlate all those because you were asked all those things. So I really want to know from you. So so you can see here that when under the low phosphate, the root length is going, the lateral root growth is happening as per the literature, and under the low sulfur, the root length is increasing. So all these story, even the photo bleaching response is on the minus. So summary of my result, whatever we did in this paper, is elongated hypocotyle directly binds with the light responsive element of swelling promoter, negatively regulates swelling biosynthesis in light dependent manner rhabdosis thaliana. So this is the model. Whenever the dark high 5 will not in functional. So if high 5 is not functional, it will not go and bind with the G box says on the promoter. As a result, squalene biosynthesis will take in a prominent way. What will happen in the light? High 5 is functional, light come into the picture, and this G box is not free, it will bind with the high 5. So if high 5 and G box is engaged, then a squalene biosynthesis will not take in a prominent way. As a result, it will hamper. So this is the first part, and when we will talk about the different the abiotic stresses. So you can see here that nutrition deficiency, both gene is working in a positive way. Temperature, both gene is working in synergistic way. In drought, both the gene is working in a contrast way. So what is my motto? Motto is very clear. So even we have checked the light and dark sample and transcriptome data in tomato and tobacco, they are showing the same type of scenario, that in dark squalene will be the higher. So can we produce some plant having already squalene is higher, and we manipulated the light and dark condition of the plant, and then can we produce higher squalene from the plant versus squalene to save our mother earth? So this is about the abiotic stress, and this is some unpublished data which where we have checked the concentration of when we applied that high amount of the squalene. So the endophytic bacteria is not able to survive at the higher concentration of the the squalene concentration. So initially I disheartened that it will not give any such phenotype in the bacterial environment. So again we optimized the 8 nanogram of squalene more than sufficient to show this response. So this is wild type and CRISPR and OX line. So you can see here that how this overexpression line shows the higher the degree of endophyte colonization into the plants. So this is for sure. So from that I will try to acknowledge my boss, current boss, Dr. P.K. Trivedi and Dr. Samir Visavant. And recently we have shown this data to the 47th annual meeting on the Plant Tissue Culture Association of India. And then this is my lab group from seniors to juniors. So now if you will give me the time, then I will discuss about your work. So this paper has nicely stated that how the Callose synthase PRM4 promotes the root hair callose deposition and adaptation to the phosphate deficiency. This is very nice story, and because I'm really new to this domain, so if I will do any mistake, then of course you have to pardon me. So Callose deposition through the glucan synthase-like GSL enhances cell wall function and limit plasmodesmatal permeability. So Arabidopsis glucan synthase-like 5 is closely related to PRM4. So contribute to the to the basal accumulation of RN induced deposition of callose in the root phloem. So what happened when phosphate deficiency, the phosphate starvation response comes into the picture, RN accumulation very high in the plants. So GSL5, that's the PRM involved in callose deposition and enhanced fungal resistance. So loss of leads to the increased resistance of pod mildew fungi through the strong salicylic acid defense response. So if PI deficiencies come into the picture, this PRM4 help the root hairs to deposit more and more callose. So if more and more callose will be deposited here, then what will happen? So it will growth will be hampered as the primary growth will restrict, but it will give the plant to stay strong during the callose deposition is act as a the strength that to reduce such as root system area reduction of anthocyanin accumulation. So this is all about different strategies. So it is like a tug of war situation. So who will win, iron or phosphate? Of course iron will be winner in this case. So this is the scenario where callose deposition will take place because iron comes into the picture. So if iron is flowing into the system, callose deposition will be the less high. So this is the story. So for my perspective, that's how this the root architecture system is being controlled by this low phosphate the low phosphate root. It's basically root architecture remodeling. Specify LPR1 help to stop primary root growth, and instead to promote the primary growth, they have helped in the secondary the lateral formation. So this so that the signal to deposit callose instead tied to the growth stunted mechanism controlled by LPR1 and 2. So plant initially it will go down, okay, so it will go down in search of water. So PI deficiency will act like in a manager that it will stop the primary root growth and it will increase the lateral growth. Root hair defective, where so here you can see here that the RDS or HD6 has no any root hair and no callose formation here. So this is the PI deficiency, another paper in which you have nicely shown that maintained or increased under PI deficiency. So you can see this PEPR1 and RLK FLS2, and this again this gene is under the negative control and this is under the positive control. So you can see here that the PERP1 has the relative abundance is higher as compared to the FLS and CRK. So this is under the negative influence and this is under the positive influence under the PI deficiency. Okay, so again stories to correlate the defense gene, because this CYP is a cytochrome 450 gene family, 71 family and the 12 number peptide. So the PI deficiency again is showing the drastic in the MAP kinase pathway system, and this MAP kinase pathway activation is going to give rise to this defense gene expression is higher under this deficient condition. So if you will see this defense gene expression, so under minus Fe condition it's increasing, and if PI is higher its expression is going down. So it means what? The pH is also influencing in the same way. That's pH, lower pH of PI deficiency is mimicking the pH iron condition also. So low pH means higher iron effect. So iron availability enhances the defense gene. So iron enhances very first step of the immune signaling. Then what will happen? This LPR1 and LPR2 mutant, which primary root growth inhibition, and then iron deficient can boost the defense gene expression to reduce this effect. So this the RB is a burst oxidase type of factor which increase the ROS. Iron amplify the immune system by triggering the ROS production. So without this the oxygen radical plant this again very nice story. So low PI leads to the iron accumulation and then so and so forth. So this is the condition. So again, so F1 expression is increasing under higher iron condition, and subsequently this WRK33 has been also is a great player in this whole story. So cut the story shorter, I will again the phosphate responsive phosphate transporters expression is also going pro attacks. So is going drastic under the low phosphates, its expression is going higher. So Pro-PEP6 is the least effective factor that you know stay the plants in a better bed. So this is the co-IP experiment. So what is happening here? If you see, this is the system. This is the pulled out experiment. This is the input. So ProPEP6 has the MAP kinase pathway, and that this PRP1 PRP2 flag has been increasing under this condition of the PEP1 is active, ProPEP6 present, and it's even PEP1 is active, then its expression is drastically increased. So all these experiment, what they are saying, that this receptor, these are the receptor, and BEC1 is a helper, this is the input. As the BEC1 is increased, this is the maximum degree of induction. Why? Because PEP1 is an input, it's a helper. So if it will increase, then the interaction is also subsequently increased. So PEP1 and Pro6 has PEP1 Pro6 is a lesion that PEP1 and Pro6 help to load the BEC1. So BEC1 is a helper, so it will handshake with this factor, and then subsequently the story is this is the pathogen, and this is the mutualist type of case. So in this case, what will happen? The oxidase, which reactive oxygen species oxidases, which specifically increase the ROS. This ROS generation leads to the PEP1 and this receptor. So BEC1 is a helper. It helps PEP1 and this factor to initiate this MAP kinase type of response, and then MAP kinase response, I think Werke 33 will winds, and this defense gene expression will be activated. In the case of mutualism, so what will happen? So this BEC1 is inhibited by this ProPEPs. So this this RBO is the reverse oxidase is not going more functional. As a result, this immune suppression will be takes place because there is no immune activation. So MAP kinase pathway is not in a function. So here the phosphate response gene, so for phosphate starvation response gene comes to the picture, and nutrition adaptation plant will take. So this is very nice story which have been published by your group, and thank you for sharing this amazing papers to read about the papers. Again, this story is again very nice story in the context of biotic prospect. So humidity, how and humidity drives ABA depletion, and how ABA depletion interacting with the stomata opening and closing, and then how calcium flux increases, and then as a result how this the plant hormone like ABA stomata open in the condition. So if stomata open, then the humidity is no longer on the surface of the leaf. As a result, the pathogen attack will be. But in the case of high humidity condition, bacterial attack happens, and then type 3 secretion system activate AVR P2B, which is a protein, a bacterial AVR protein, which induces what NCTs, which is ABA biosynthesis gene factor. So as a result, this CYPs is blocked. So CYPs is not in action, so ABA depletion not happens. ABA is higher in the stomata. As a result, what will happen, stomata will be open and water soaking condition is there, and then pathogen will spread nicely into the system. So this is all about the paper. If you will tell me, then I will explain the whole paper. So I have, these are very nice story which I have, you have given me, and I'm very thankful for giving me such environment so that I can read more and more paper. So if you will tell me, then I will explain all these things. So this is all about from my side in this case, because this paper basically it's very nice story. I have gone through this paper that you have used the different rice variety and different the high fertilizer, no fertilizer condition, and pesticide free environment, and then you have checked the nitrogenase and if how this nitrogen fixation is taking place in the plant in the field, and how this. So here is a lot of studies about because it's very less time for me to grab all those things, because several I'm associated with several projects here, and I have to commit several things before leaving India. So this paper really very interesting. I will go deeper to study well, because right now I have no any the diversity related project. I haven't gone through any diversity experiment here. So this paper is really nice. Then I think I have to study well that paper. So thank you, and I will not go into the detail of this how assay and salicylic acid biosynthesis. This is only for our students, because I always like to teach students. So if I have time, I will go through these all, how this pathogen attack, successful attack, unsuccessful attack, how this chemical defense strategy, so what plant will take and give the responses. So it's very nice story to tell you guys. So thank you so much for listening, and I think I will explain in the later.